Hi everyone, I'm Jeannie Chowning and I'm the Senior Director of Science Education at Fred Hutch. Welcome to my kitchen. And is everybody ready to do some DNA extraction today? Great, this is one of my favorite activities and I'm so glad that you'll be joining along with me. So first off, I want to thank you for participating in Obliteride. It's a really special event for Fred Hutch and we really appreciate your participation. So we're going to try and get DNA out of a strawberry. And so here are the different things that we're going to use to get DNA out of a strawberry. We have the strawberries. You can use frozen strawberries. We have water. We have about half a cup of water. The exact amount's not important, but about half a cup. I have another cup that I'm going to make my DNA extraction mix in, and I have a plastic teaspoon. I've got some dish soap. I've got some pineapple juice. Meat tenderizer plays the same role here. If you don't have this ingredient, don't worry about it. This is the least critical one for what we're doing today. And then I have an empty cup for our strawberry DNA mix. This is where it's going to end up in here. And I also have some salt, just regular salt. And then I have a wooden stick. In my case, I'm using a chopstick. I also have some cheesecloth. You can also use a coffee filter if you don't have cheesecloth. And I just fold it over once. You don't want to make it too thick. Okay, so let's get started. We're going to try to use a strawberry to get the DNA out of the strawberry cells. And you might have heard that all living things are made up of cells. And if I look close enough at my hand or at the strawberry or at the leaf on my plant over here, I would see cells. And inside is the DNA, which is the instructions that the cell needs to do its job, to grow, to develop. And so it's pretty important. And one of the things that Fred Hutch scientists do is study DNA when we're trying to fight cancer. So to try to understand cancer better, to make new drugs. And when scientists isolate DNA or take out DNA, they do pretty much the same thing with just like a little bit of difference, but the same idea. So it's a pretty cool process. So we're going to start with 2 strawberries, and we're going to take off the leaves. We don't need the leaves. So take this. This is actually my favorite part, is this step. We're going to put them into a resealable plastic bag, and we're going to seal up the bag so the mush doesn't get out. And then we're going to mush them up. And this, I think, feels really cool. I enjoy doing this part. So just go ahead and mush them, and we're going to mush for about a minute or 2 until it's pretty evenly mushed out and you don't have a lot of big pieces. Like if you have some clumps, that's okay. We're just trying to get a good even mush here. Doesn't this feel interesting? Okay. Strawberry mush. What we're doing here is breaking apart the cells that make up the strawberry, and especially trying to break up the cell walls. And the cell walls are on the outside of the cells in plant cells. And so using some pressure here and breaking them apart is our first step in getting the DNA out. Okay, I think that looks pretty good. Next, we're going to make our DNA extraction mix, and I'll show you what goes in there. And I'll tell you what each part does. The water just helps make everything mix up together, and so we're going to pour the water into the cup here, and I'm going to take 2 teaspoons of dish soap. Cells also have layers in them that are made up of kind of a fatty substance, and the soap helps break up those layers so that we can get the DNA out. Okay, there's a membrane on the outside, or a layer on the outside of the cell, and then another layer right outside where the DNA is stored. And so the soap is going to help us break those layers apart. Good. Now we're going to use a teaspoon of salt. And the salt helps with the DNA clumping, because the DNA is actually negatively charged, and if we didn't have the salt, it wouldn't clump together. It would kind of stay apart. And so the salt breaks up into positive and negative charges when it's in water, and the positive charges bind to the DNA, bind to the negative DNA, because opposites attract, and that helps the DNA clump because it's not charged anymore after the salt is interacting with it. So it's helping us with the clumping. Okay, so I want to mix that up just a little bit. And then the pineapple juice is going to provide a chemical that's going to break apart some of the other pieces of the cell that we are not interested in. So the chemical is an enzyme, and it breaks apart some of the proteins in the cell. So we're just going to give a little tiny bit. And meat tenderizer, you just use a pinch if you have meat tenderizer. Again, don't worry if you don't have that. I'm going to just mix that up gently. And then I'm going to open my bag again. And I'm going to put in 2 teaspoons of my extraction mix. I'll take 1, 2. Okay, and then seal it back up again. And we're going to mush again, but this time we really want to make sure that we're not mushing too vigorously, because we don't want to make a lot of soap bubbles. So we're going to mix kind of gently here, so we get this, our extraction mix all mixed up with our strawberry mush. And so just going to spend about a minute gently mixing those things up. Okay, how's everybody doing? Hopefully get your soap mixture mixed in here, and we're gonna next filter out all this other stuff, parts of the plant cells that we don't need, especially the big parts. Like I can feel like I have a big piece of the strawberry in here that we're not gonna need, and we're gonna put that into our strawberry DNA final cup. So here I'm going to put 2 layers of the cheesecloth together, or use your coffee filter, and I'm going to make a little indentation here. And now I'm going to transfer this substance into the cup that has the cheesecloth. Okay, kind of pour it out gently. And you don't need to get every single bit of it. If you can't get every single bit, there's still plenty of DNA here that we'll get to see. Okay, so I'm going to lift this up. You can already see some liquid coming out. I'm going to just squeeze it, close the top a little bit, and squeeze gently to get the liquid to come out. So I'm just going to do this for a few more seconds, so I get a nice amount of liquid. Squishing it out. Can't wait to see if we got DNA. Okay, the last step requires cold alcohol. So hopefully you have some cold alcohol. It could be rubbing alcohol. I'm going to use an alcohol called ethanol. And I'm going to get it from the freezer. It's actually okay to be in the freezer. And then I'll show you what to do next. Okay, here, the whole thing we want to do is make a layer of alcohol on top. So we don't want to just pour it right in the middle. We want to pour the alcohol down the side of the cup gently, so it just very gently sits on top of the strawberry mixture, because what's going to happen to the DNA is it's going to show up in the layer in between the alcohol and the strawberry mixture. And what happens is that DNA doesn't mix very well in alcohol, so it's going to come out of the strawberry solution, and you'll be able to see it right at the interface between. So I'm going to tip my cup a little bit so I can run the alcohol down the side very slowly. And you want to add about as much alcohol as you have strawberry mixture. So that looks good. Maybe a little bit more. Okay. And so what I'm seeing is a bunch of bubbles and a bunch of stringy stuff. And what are you seeing? You seeing anything happening here? I'm going to get my stick and I'm going to gently move this around here. And I can see that I have a whole bunch of gloppy white stuff. What does yours look like? Hopefully you have some gloppy white stuff too. And here is a whole bunch of DNA. And you can see that it's really kind of stringy and gloppy. And strawberries have a lot of DNA in them. So hopefully you got some DNA out of your strawberries too. That was a lot of fun. Thanks for joining me, and I'll be back a little later with our next activity. Hi everybody, welcome back. Are you ready for our second activity? Let me show you what we have here. We have Q-tips, we have dish soap, we have some food coloring, and if you don't have food coloring, you can use pepper. And I have a little square of watercolor paper. You can use cardstock if you have it. Printer paper doesn't work so well. Then I have a shallow dish that I can use to put the milk in. And if you don't have that, you can use a plate. So I'm gonna go get the milk, and I'll be right back. I'm using whole milk, works best with whole milk, but if you don't have whole milk, 2% will do. And so the first thing you do is add the milk, and you want about 1/4 of an inch, so not too much, just kind of covering the bottom like that, and then just let it settle for a bit while you get your other materials ready. So I'm going to do the food color next, and I'm going to put the food color right in the middle, and I'm going to put the drops close to each other. So here we go. A lot of colors is good if you have a lot of colors. So I'm going to put 1 drop there. Actually, let me do 2 drops. And a couple drops of red. And here's a darker green. And then a blue. And then a kind of pink I found. All right, and so we've got our drops in the middle, and now we're going to take a Q-tip. So the first thing I want you to do is take a Q-tip out, and we are going to touch it right into the middle of the colors. And I want you to think, what do you predict is going to happen? What do you think is going to happen? Okay, so think for a minute, make your prediction or your guess, and now let's do it. All right, you see a little something there, but not a whole lot. Now I'm going to flip that over and stick the Q-tip into the soap. And then I'm going to hold it above here and do the same thing, but before you go in, predict again. What do you think is going to happen now? So I'm going to put it in. Whoa! I'm going to hold it there for about 30 seconds or so, and you can see like a lot of motion. If you look in the middle, look at the movement. And the colors are just moving like so fast. And this goes on for a while, and you can kind of watch it until it stops, but mine is still going. The colors are looking really nice. I hope yours are too. Now I'm going to try something that I heard about but I've never done before, so this is going to be brand new for me too. I'm going to take the piece of paper and I'm going to put it over and kind of press down a little bit. And I'm going to take it out, and wow, that's really beautiful. You get some colors that stick on the paper, and I'm just going to put it over here and let that dry, and that will make a nice card for somebody. And you can see the colors still mixed up there. So one of the things that you can think about is, what did you observe and what questions came up for you? That's a really important part of science, is looking carefully at what happens and what kinds of things you see, and also asking questions about them. Like, what do you think is going on there? Another thing you could do is think about what else you could try. So we used whole milk. What would happen if you used non-fat milk? What would happen if you used water? There are a lot of different things you could try to see if you would get the same effect. And I encourage you to do that. Just experiment and see what you find out. So this activity is actually connected to the importance of hand washing. And I wanted to talk to you just for a moment about why that is true. So if we could zoom in really, really close to milk, what we'd see is globs of fat in the milk and lots of water around the fat. Especially if you've got whole milk, it's got a lot of fat in it. And so if we zoom into the soap, we'd see something that had like two different ends. Like one end that wanted to bind to soap, one end that wanted to bind to fat, and another end that wanted to bind to water. And so what happens is the soap molecules try to get at the fat that's in the milk, and to have the end that likes to bind to fat bind. And so it's moving to try to encircle the fat with soap. And so when the soap is surrounding the fat, it helps break it up. And that's why we wash dirty dishes with soap. But it's also why we wash our hands with soap. So this is kind of a picture of a virus. And the outside is kind of a fatty layer here. And if you use soap, the fatty layer will break up, and then the virus will fall apart. And so this is actually why washing hands is super important when we're trying to be careful and not have viruses on us. It's even more effective than hand sanitizer because you're breaking up the outside. So I hope that this activity was fun for you. If you have a colorful piece of paper, why don't you hold it up and show everybody? And I'd love to see what you did and how yours came out. Okay, thank you.